Hello students, I am Akshay Deshpande and welcome to the biology class. And today we will be looking into the class 2 of biomolecules. And in this particular class, we will be mainly concentrating on the primary metabolites and secondary metabolites. We will also look into the concept of biomacromolecules, that is, on what basis we say any molecule is a large molecule. Okay, the term macro mainly means large molecules, right? The term biomolecules mainly means they are the large molecules, right? Yes. So on what basis we can say that these molecules are large? And also we will look into one such category of biomacromolecules, that is the polysaccharides, right? So polysaccharides actually belong to carbohydrates. What we are looking over here is the carbohydrates. So I think we can start our discussion on biomolecules. So now we are having lots of chemicals in the living organisms. Okay, so we generally say that one of the important and exciting aspect of the chemistry is mainly isolating of these thousands of chemicals that are mainly present in the living organisms. Okay, mainly in the living organisms. We try to isolate all those chemicals, right? And also we have to determine, we will try our best to determine the structure of these chemicals, right? So now why do we need to isolate the thousands of chemicals or try to determine the structure of these chemicals? It is quite simple, because of their economic importance. We can say that they are quite economically important, and some of them are also important in the medicinal terms. Okay, also in the medicinal terms, some chemicals are also quite important, right? So by determining the chemical property, right, and also their structure, and if we can even try to synthesize them artificially in the laboratory. Okay, so this is going to be a great breakthrough, right? So in many cases, in many cases, what happens is that plant may be the source. Plant may be the source of chemicals, right? Yes, the plants are the source of the chemicals, and the chemicals which are present in the plant body, or you can consider any living organism. Okay, you see that these chemicals are actually present in a very scanty amount. Okay, very scanty amount. So now what do we mean by a scanty amount? Very less quantity, very less quantity, right? So now as they are present in less quantity, if we have to synthesize, if we have to require them, if we happen to require them in a large quantity, so we have to go and scale up lots of plants. Okay, we have to identify lots of plants, or in some cases we have to go for plantations and all, right? And by planting the plants or the trees into the saplings of that particular plant in the plantations, and we have to wait for a long period of time until they actually produce that particular chemical, right? So we can actually avoid this method. Okay, and what we can do is we can directly extract the chemical. Okay, we can directly extract the chemical, and by extracting the chemical we can even determine the structure. Okay, we can determine the structure of that particular chemical, right? And also if we can determine the structure, we can look into the bond arrangements. Okay, so the structure will mainly tell about the molecules which are present in that particular chemical, and also we can determine the bonds, right? What kind of chemical bonds we are mainly speaking of, right? Chemical bonds, right? So what kind of chemical bonds are mainly present in that particular chemical, and by determining the structure of all that chemical present in the plant, what we can do is with this particular knowledge we can actually synthesize we can synthesize the chemical, right, in the labs. And through the course of time, what we can do is we can purify it, and we can even increase the affinity, or we can even increase the efficacy, the effectivity. Okay, so this method will result in increase in effectivity, right? The drug will become more effective, right, if we try to synthesize them artificially in the lab, because we can arrange the bond however we can want based upon the natural molecule that has been found in the plant, right? So in this way, we can see that there are thousands of chemicals which play a very important role in day-to-day of our life, which can be derived from the living organisms. Okay, so in major of the cases, the main sources of these interesting chemicals are mainly from the plants, and in some cases it is even the fungi, right? Even fungi, that is mycota, okay, even they also have certain chemicals. For example, antibiotics is a very interesting chemical that is produced from the fungi. And even the microbes, right, even the microbes do exhibit or do produce certain chemicals, right? And these certain chemicals are having human value or economic importance for the humans, right? So hence the main aim of the chemistry is to determine their particular structure and try to synthesize them artificially in the laboratory. So now in the living organisms, okay, we have discussed in the last class, in the case of living organisms, the organic compounds, we call them as the biomolecules, right? We call them as the biomolecules, right? Any carbon derived structure is called as an organic molecule, and this organic molecule in the living system we call them as biomolecules, right? And these biomolecules are called as the primary metabolites. They are called as the primary metabolites. Now, the reason why they are called as primary metabolites is because they are having some cellular function. They play an important role in the cell, right? So they may be involved in the metabolism, right? They may be involved in some anabolic reaction. They may be involved in some catabolic kind of reactions, right? So we can say that they do have certain important functions, right? They do have certain important functions in the cell body, right? So we have named various biomolecules in our last class. For example, we are having amino acids, right? We are having amino acids, and we are having carbohydrates, right? Yes, we are having carbohydrates. We are even having the proteins, right? Yes. And also we are having various others such as the lipids, right? Yes. Nucleic acids, right? Yes. And that is the nucleotides in the case of nucleic acid, nucleotides, right? And amino acids in the case of proteins, right? And carbohydrates in the sense we are mainly speaking of simple sugars, right? So we can say that these are actually called as the primary metabolites: the amino acids, the lipids, the nucleotides, which are mainly involved in the formation of nucleic acids and all. All of them are actually called as the primary metabolites because they are having certain cellular functions. Okay, they take part in certain or one or the other cellular activity. Okay, hence we call them as primary metabolites. So we can say that primary metabolite, okay, primary metabolite is something whose function is known, right? Primary metabolite is any substance or any chemical or any organic molecule that is present in the plant body or any living organism's body whose function is actually known, and they take part active role in these cellular processes, right? Hence they are called as the primary metabolites. But in certain occasions, especially in the case of organisms such as the fungus, okay, fungal, and also in the case of plants, and also we are having microbes in the sense bacteria, what we are speaking of, right? So in the case of a fungi, in the case of a plant, and also in the case of microbes, you do find certain substances called as the secondary metabolites. Okay, now secondary metabolites are mainly whose functions are not known. Okay, their function is not known. Their function is actually not known, right? So what do they do? We don't know yet, but yet they are actually present in the body of that particular organism, right? So we can say that these chemicals, okay, they are not only few in number, they are thousands in number. Okay, thousands of we can see such chemicals are present in the body of the plant, microbes, and also in the case of fungi, right? So the metabolites or the substances which are present in the living organism's body, and yet we don't know their function properly. Okay, what role they play in the organism's body, we don't know yet. Okay, but however, one of the important idea that we do have regarding the secondary metabolite is that they play defense mechanism. Okay, they play an important role in the defense mechanism. Okay, defense role, right? See, in the case of plants, okay, in the case of plants, we see that many substances, for example, we are having the alkaloids. Okay, we are having alkaloids. Alkaloids are known to play a very important defense mechanism in the case of plants. Why? Because plants, unlike the organisms, okay, unlike the animals, they cannot move from one particular place to another place. They are fixed. Okay, they live in only one particular region. They cannot move from one place to another, right? So there may be threat from the herbivores. Okay, herbivores are the organisms which mainly feed on the plants, right? So some of the plants exhibit a defense mechanism by producing the alkaloids, and the alkaloids mainly give a toxic or an obnoxious feeling to the plants when the particular feeling to the herbivores when they feed on the plant, right? As a result, we can say that the herbivores try to avoid such plants which are having pungent or repugnant chemicals present in their body, right? So alkaloid is one such example. The next one we are having is flavonoids. Okay, now flavonoids are excellent coenzyme molecules. Okay, you will study about the flavonoids, and also one more, that is the NAD. Okay, NAD plus. You will study about that. It's also an example for electron carrier, right? So flavonoid, we are having example. The FAD is an example for FAD is an example for the flavonoids. Remember, yes. The flavonoid, one example we are having is the FAD, and you people might have heard about FAD in the chapter that is the photosynthesis, and also in your respiration chapter, right? So they are taking place an important role in the metabolic reactions. Okay, and in the case of plants or fungi, they may be byproducts. And we are having one more, that is a rubber, and rubber is actually a polymer, right? So we all use the products that are derived from the rubber. Okay, rubber is actually a latex kind of material which is derived naturally from some of the trees, right? And in some cases you may find some essential oils. Okay, the essential oils play an important role in a regular cooking activity, or they may play an important role in the traditional herbal medicine also. Okay, essential oils. And the most popular, we are all very much aware of, is the antibiotics, right? Now in the case of antibiotics, these are mainly produced by the fungi. Okay, remember. Okay, mainly the fungi produces the antibiotics, and antibiotics, they are mainly playing an important role in the defense against the bacterial growth. Okay, so the fungi mainly produces the antibiotics to inhibit or to prevent the bacterial growth surrounding them. Okay, so this is an a product that we get from the fungi, and we classify or we consider antibiotic as a we can say a secondary metabolite, right? And also you find pigments, right? Now pigments are, generally in the case of flowers, right? Yes, the flowers mainly produce the pigments, right? Yes. So now these pigments in some cases, the pigments may have antioxidant activity, right? Antioxidant activity. Now, if you try to analyze what do you mean by antioxidant activity, see, during the cellular reactions, in the cell there are reactions, right? Yes, we all know about the cellular reaction, that is the respiratory kind of reactions, photosynthetic kind of reactions, right? And generally there are some species of radicals that are formed, okay? ROS, okay? The ROS mainly stands for reactive oxygen species, okay? Reactive oxygen species. And the reactive oxygen species may cause a damage to DNA, okay? They may cause damage to the DNA, right? So these antioxidant substances such as the pigments, what they can do is they can give an electron to the ROS, right? They can give an electron to the ROS and they can stabilize or they can nullify the activity. They can nullify the activity of the ROS, and which can prevent any kind of risk or any kind of cellular damage that can be given by the ROS, right? So antioxidant activity is observed in the case of pigments, okay? And one such pigment that which is known to have antioxidant activity we are having is the anthocyanins. Yes. Now anthocyanin is known to have an antioxidant activity, right? So now we are having one more that is called as the gums, okay? Gum is also very much what we can say similar to the rubber kind of material, and scents are mainly some sticky substances which are known to produce some good odor, okay? Pleasantful odor that we generally use the scents in the case of manufacturing or synthesis of perfumes, okay? We do apply perfume on day-to-day basis. So, and finally we are having gums and resins, okay? We are having gums and resins, okay? Now resins are mainly gum-like substances, okay? And these gum-like resistance, what we can say, substances, they are mainly involved in giving some polymeric or sticky kind of property, okay? Resins is one example, and we are having one more, that is the spices, okay? Spices is also we can say it is also an example for the secondary metabolite, okay? So as we have mentioned, their functions and roles are not yet known, okay? What do they do in the plant body? We have no idea regarding that. As of now, we don't know, or we don't have any kind of idea. But however, we have a list over here. Let me erase this. Yeah. So we are having a list of substances, okay? Mainly the secondary metabolites, right? Yes. We are having some mentioned secondary metabolites, okay? And their broad category, okay? The pigments, alkaloids, terpenoids, right? And these are all actually, we can say, the category, and we are having the examples of that particular category over here, right? So we will, this chart is again important. Remember, this chart is important because there have been multiple questions appeared regarding each of these constituents, okay? They have asked all of these what we can say the category of that particular secondary metabolite and what is an example to that particular category, okay? So don't worry about the functions. They have not mentioned any functions in your textbook, so you don't have to bother about it much, right? But however, I will try to give an some functions of certain secondary metabolites over here. So the first one we are having is pigments, right? The first one we are having is what? Pigment. And an example for pigment we are having is the anthocyanins, and one more we are having is the carotenoids, right? So now in the last class we had mentioned about the carotenoids, right? And in a few of the last classes we have mentioned of the carotenoids and xanthophylls, if you remember, okay? And beta carotene, they mainly belong to carotenoids, right? And generally the carotenoid is rich in the case of carrot, right? You know, the carrot is known to have known to have high amount of carotenoids, and we, generally many elders suggest to young children to eat more carrots because it is good for the health of the eye, right? Yes, because carotenoids mainly contain the precursor molecules or starting molecules that are required for the synthesis of the retinal. Okay, yes, retinal. Okay, you will study about the retinal and mechanism of eye vision in the case of a human physiology, that is nervous system, right? Yeah. And just a moment ago we mentioned about the anthocyanins, and we all know anthocyanins mainly exhibit what kind of property? Antioxidant activity, right? We mentioned about this just a moment ago. And similarly we are having one more, that is the alkaloids. Okay, so alkaloids are generally a substance that are mainly stored in the stem of the particular plant and also in the leaves of the plant. And one such alkaloid we are having is the morphine. Okay, the morphine, okay, if you remember morphine, you people might have studied this in the human health and diseases for the drug and abuse part of the chapter, right? So now morphine is an excellent painkiller, right? Morphine, okay, morphine is an painkiller, right? It is also called as analgesics. So it is an excellent painkiller, morphine. But however, many people abuse morphine as a drug. Okay, so many people take morphine as a drug. You also see that it is abused, drug abuse. It is also an example, morphine. And similarly, similar to the morphine, we are having one more that is called as the codeine. And codeine is also a kind of analgesic, which is mainly used to treat the pain and other associated, mainly fever, pain, body ache and all, right? It is mainly used, it is treated by the codeine, right? Now the next one we are having is the terpenoids. Okay, now terpenoids are complex oil-like structures. Okay, they are completely complex oil-like, and they are mainly organic substances. Terpenoids, they are mainly organic. Yes, and they are used to synthesize certain organic chemicals. They are used to synthesize a certain organic chemicals, right? And based upon the simple structure, we can call them monoterpenes, and second is diterpenes. See, you can easily identify the presence of the terpenoids in the plant if you take a section of a stem of a plant. Okay, if you take the main stem of the plant, you can see that in the case of terpenoids, if you take a section of a stem, okay, yes, the center, you see that generally it is dark shaded, right? The center is quite dark shaded, and that is mainly because of a deposition of terpenoids. It is mainly because of deposition of the terpenoids. And we are having some essential oils. One example that they have mentioned for the essential oil we are having is the lemongrass oil, right? The lemongrass oil is used for various, what we can say, traditional medicine, and also it does have some medicinal importance in the ancient Ayurvedic system of medicine also. Now we are having a toxins. Toxins are nothing but the poisons. Okay, and we are having two examples. The first one we are having is the abrine, and one more we are having is the ricin. Okay, now abrine, okay, abrine is quite toxic. Okay, they say that only 0.7 μg is sufficient to per kg, right? μg per kg. Yeah, this is what they use, standard terms. μg per kg is sufficient to kill the mice. Okay, one mice can be killed by just 0.7 μg per kg, right? And in the case of humans, it ranges from around what we can say, around 20 to 20 μg to 30 μg, right? At least that is what they say, per kg is sufficient to kill the humans, right? So they say that the abrine, and one more we are having is the ricin, right? Yes, ricin. Abrine and ricin, these are one of the naturally known highly toxic substances. Okay, naturally known highly toxic or high toxins, we can say, right? So they are highly toxic to the human body. Similarly, resin, okay, and in the case of resin, they say that around 10 to 1000 mg per kg is sufficient to kill the humans, right? Around 10 to 1000 mg per kg is sufficient to kill the humans, right? So they are mainly toxins. Aberrin and resin, they are mainly toxins. The next one we are having is the lectins. So see, lectins are mainly some carbohydrate molecules, sugar attached molecules. Okay, yes. And one example for lectin we are having is the concanavalin A, right? Concanavalin A. Now it is used as a drug. Okay, it is mainly used as a drug, mainly to treat what we can say, cancer. Okay, it is mainly used to treat cancer. What kind of cancer, if you ask? It is mainly used to treat the liver cancer. Okay, it is mainly used to treat the liver cancer, hepatoma. We also call it as what? Hepatoma. And there are some drugs, okay, okay, and the example for drugs we are having is the vinblastine, and one more we are having is the curcumin. Okay, now vinblastine is again used to treat cancer. Okay, vinblastine, right over here, vinblastine, right? It is mainly used to treat cancer, right? And similarly, curcumin is also an important drug that is derived from the turmeric. Okay, it is mainly derived from the turmeric, and even the curcumin is also showing, also exhibits the anti-cancer kind of properties, right? Vinblastine and curcumin are having important function in treatment of cancer. And finally, we are having a certain polymeric substances. For example, rubber, it is a latex, as we all know, and we are having gums, okay, which is similar to rubber, okay, and they mainly provide the sticky and the stretchability kind of property if we add these gums into any kind of substance if we are required in. And finally, we are having the last one we are having is the cellulose. Okay, cellulose, we are all very much aware, it is a polymer of what we can say, glucose, right? And the cell wall in the case of plant is mainly made up of the cellulose, cell wall, right? So these are some of the important what we can say, the substances or the what we can say, secondary metabolite substances and their respective examples with a certain function that we know. These functions we have determined now. Okay, so earlier when they were discovered, their functions were not known properly. Okay, so similarly, imagine there are thousands of such kind of chemicals produced in the plant body, fungal body, and also microbial body, and determining their roles and their function would be having a lot of human benefit, right? Now moving on, we are having the next point, that is the biomacromolecules. Okay, biomacromolecules. Now the biomacromolecules, why do we call them as biomacromolecules? Because they are large in molecular mass or molecular weight, right? Yes. So now they say that they have been mentioned that you see that there are two types of fractions. The first one we are having is the acid soluble fraction. Okay, if you remember from our first class, we had determined the main important substances by grinding the cells or tissue into the trichloroacetic acid, and we got two types of fraction. The first is called as the acid soluble fraction, and one more we are having is the acid insoluble fraction. That is what we determined, right? So now generally the acid soluble pool is having the molecular weight of around 18 to 800 daltons, right? Any substance or any chemical or any molecule that is present in the acid soluble pool, it is known to have a molecular weight of around 18 to 800 daltons, right? And similarly, the acid insoluble pool mainly includes the proteins, polysaccharides, nucleic acids, and the lipids. Okay, these are mainly belonging into the acid insoluble pool. Okay, now any biomolecule, they have mentioned, right, any biomolecule whose mass or whose molecular weight is less than the 1000 Daltons, we refer these molecules as the micromolecules. We refer them as what? Micromolecules, right? So in other terms, any biomolecule, right, is having more than 1000 Daltons of mass, then we can refer them as the biomacromolecule, right? Yes, this is what we can determine by the term biomicromolecule and biomacromolecules. Okay, if the molecular weight is less than the 1000 Daltons, then we refer them as the micromolecules, and if the biomolecule molecular weight or mass is more than 1000 Daltons, then we can refer them as the biomacromolecules, right? So roughly speaking, if you have to identify the biomacromolecules, okay, protein is a biomacromolecule, polysaccharide is a biomacromolecule, nucleic acid and so is also lipid, we call them as what? Biomacromolecules. But however, there is an exception to this particular rule, and that is mainly the lipids. Okay, lipids are very interesting in nature, because we say that their molecular weight, their molecular weight or mass is not more than, okay, is not more than 800 Daltons, right? Their molecular weight is not more than 800 Daltons, right? But however, just a moment ago we said that any molecule is having more than 1000 or thousands of Dalton as the molecular mass, we say them as what? Biomacromolecules. Then why do lipids are exception to this particular rule, right? And they also appear, okay, not area, appear. They also appear in the acid insoluble fraction. Okay, so now when you grind the tissue with the buffer, that is the trichloroacetic acid, we observed that not only nucleic acid, polysaccharides and proteins, even lipids also appeared in the acid insoluble fraction. Okay, so now in the acid insoluble fraction, we also mentioned that all of the molecules are having more than 1000 Daltons of molecular mass. But lipids, you have seen that it is less than or doesn't even cross 800 Daltons, right? Now, the reason why the lipids actually appear in the what we can say acid insoluble fraction of the pool, because lipids are the major constituents in the cell membranes, right? In the cell membranes, right? Yes, lipids, though they are present as lipid molecules in the cell, but they are the major constituents in the case of cell membranes. So what happens is that grinding the tissue basically breaks the cell membrane, breaks the plasma membrane or the cell membrane, all mean the same. Okay, it basically disrupts or breaks the plasma membrane, right? So now what happens as a result of breaking of plasma membrane is that the lipids which are mainly present in the cell membrane, they mainly form the vesicles. Right? Vesicles are, as we all know, in the cell, they are small bubble-like structures. Right? These are called as the vesicles. And these vesicles actually appear in the acid soluble pool. They actually appear in the acid soluble pool. Okay, and they are also, as we know, lipids are mainly water insoluble. We also know that it is water insoluble. right? Yes, lipids are completely water insoluble in nature. As a result, what happens is that they mainly appear in the acid acid insoluble pool. So, strictly speaking, we can say that the lipids are not macromolecules. Okay, I just made a mistake. It is not acid soluble, it is acid insoluble pool, right? It is mainly acid insoluble pool. So, grinding the tissue mainly breaks the cell membrane or the plasma membrane, and not only a lipid is present in the directly cell membrane or plasma membrane, also the membraned organelles. Even in the case of membrane organelles, right? Even in the membrane organelles, you also see that lipid is one of the major constituent, right? So grinding the tissue breaks the plasma membrane and also the membrane constituent of the organelles, and they basically result in the formation of small vesicles, and these vesicles, as they're completely made up of lipids, they are water insoluble, and they precipitate or they finally are formed in the acid insoluble pool. Okay, yes, so it is mainly acid insoluble pool. So strictly speaking, we can say that based upon the molecular mass, okay, if you consider the molecular mass, the mass is around 800 daltons. Okay, so from the mass perspective, we can say that lipids are not biomacromolecules. Remember, okay, from the molecular weight or molecular mass perspective, we can say that lipids are not actually macromolecules or biomacromolecules, only because they are water insoluble, and grinding results in breaking of cell membrane, and they precipitate or they result in the formation of what we can say vesicles, and they finally land in the acid insoluble pool. Okay, that is why we can say that lipids appear in the acid insoluble pool. This is an explanation that we have to remember for our discussion. Now, if you take the cell, if you consider the cell, okay, we mainly find two types of constituents. The first one we are having is the cytoplasmic constituents or cytoplasmic components, and one more we are having is the organelles and also the macromolecules. So now in a cell, the cytoplasm, we call it as the cytoplasmic constituents is mainly referred as the acid soluble fraction or acid soluble pool. But whereas the macromolecules and the organelles, we call them as the acid insoluble pool, right? So they may ask you the question stating that which of the following cellular component or constituent is actually acid soluble or acid insoluble, right? It is the macromolecules and the organelles that belong to the acid insoluble pool, and the acid soluble pool mainly consists of cytoplasm. Yes. So now here we are having another table that basically shows your what we can say the components inside the cell, okay, and also what amount or what percentage they are found in the cellular mass. Okay, what is the contribution, what is the percentage of contribution they are found in the cellular mass, right? So for example, if you consider water, okay, water, we all know ultimately cytoplasm is made up of water itself, okay, and it is around 70 to 90% of the constituent. Okay, in a cell, around 70 to 90% is completely the water that you find. And the next one we are having is the proteins. And as you can see, the proteins are the next major constituent present in the cells. Okay, they contribute around 10 to 15%, and carbohydrates contribute around 3%. Okay, and lipids contribute around 2%, and 5 to 7% you see that it is mainly the nucleic acid, that is the genetic material, DNA and RNA. And finally we are having ions. Okay, so here if you have to choose which of the following is acid soluble and acid insoluble, you can say that water and ions are actually acid soluble, right? They're mainly acid soluble. And finally proteins, lipids, carbohydrates and nucleic acids, we can refer them as acid insoluble pool. They mainly appear in the acid insoluble pool. So this is quite a clear concept. Okay, so again you may expect the match the following kind of question where they may tell about the component and you have to match with respect to the respective percentage. Although the question has not appeared based upon my observation, mainly on this part, but remember, you have to remember this. It is an important table that you have to remember at any cost. Okay, as I said, try to go with the component and the table as much as possible. Write and practice. That is the best way. Right. So now let us look on to the next one. That is the polysaccharides. Let us look into the polysaccharides. Now, polysaccharides, the term poly means that it mainly means many, and the term saccharides mainly means sugar. Okay, and polysaccharides are actually group of carbohydrates. Remember. Okay, now carbohydrates, we can classify them into mainly three basic categories. We classify carbohydrates into three broad categories. The first one we are having is monosaccharides. Okay, monosaccharides. And next one we are having is the oligosaccharides. Right? And finally we are having the last one, that is the polysaccharides. Yes. So again, mono means single sugar, oligo means 2 to 10 sugar units will mainly make us the oligosaccharides, and if it is more than 3 or 4, okay, continuous, more than 3 to 4 and 10, we call them as what? The polysaccharides. Okay, so though we will not be discussing about the monosaccharides and oligosaccharides, okay, we will be mainly concentrating on polysaccharides. I just thought of mentioning the basic classification of the carbohydrates. So now, polysaccharides, as I said, they are mainly made up of many multiple sugar units. Hence, they are called as the polysaccharides. That is what they tell us here. Long chain of sugars, that is the monosaccharides. Okay. So what happens is that if I take one sugar, okay, and link it with another sugar, and similarly I will add one more sugar, and similarly I will add another sugar, and so on, we can say that this is actually a polysaccharide. Okay, what you're looking over here is simple linking of one sugar with the another will result in the formation of what? Polysaccharides. And in most of the cases, general of the cases, you see that polysaccharides are generally homopolymer, right? They are mainly homopolymer. Now, what do we mean by homopolymer? Okay, generally we are having two kinds of terms. The first is called as the heteropolymer, okay, and one more we are having is the homopolymer. Homopolymer means that if they are made up of identical sugar units. Okay, if they are made up of identical sugar units, it means that for example, let me take one sugar, that is the glucose. Okay, I will link one more glucose. Okay, similarly I will link one more glucose, another glucose, and so on, right? So if you consider this polysaccharide, you can see that it is just made up of glucose, and so on with n number of glucose, right? So such kind of polysaccharide which is made up of a same or a similar or identical sugar units, we call it as the polysaccharides. Right? Hence, in most of the cases, major of the cases, as I mentioned, the polysaccharides are a homopolymer. It doesn't mean that you don't find any kind of heteropolymers or heteropolysaccharides. You do find. Okay, if you have to give an example for a heteropolysaccharide, we are having a chondroitin. Okay, chondroitin is an example for a heteropolysaccharide. Heteropolysaccharide, right? Yes, it is an example for a heteropolysaccharide. It is made up of different units of sugars. Okay, it is not made up of single identical monosaccharides, right? Yes. So thus we can say that they are mainly homopolymer in nature. And here we are having examples. That is, first one we are having is the cellulose, okay, which is mainly made up of glucose, okay. And two more examples which are similar to cellulose in the sense mainly with respect to the sugar unit that is mainly used, okay, starch and glycogen, okay, starch and glycogen. Now remember, starch and glycogen are also similar to cellulose, but the main difference is that the starch is mainly used as the storage material, food storage material in the case of plant cells, and glycogen is mainly used as a storage material in the case of the animal cells, okay. So broadly speaking, we can say that cellulose, okay, it is mainly present in the plant, okay, cell wall, and starch, okay, it is mainly used as a food storage, okay. And finally we are having is the glycogen. It is also similarly in the case of food storage, okay. But however, this food storage is observed in the case of animal cells, right? This is mainly used in the case of plants, mainly used in the case of what? Plants, right? Yes. So now all of these three, what you're looking over here is actually used the monosaccharide that is used for in the cellulose, starch, and glycogen is the glucose. In all of these three polysaccharides, the monomer, that is the sugar unit that is found, is the glucose, right? Yes. But however, what makes a cellulose different than the starch, and what makes a glycogen different than from the starch and the cellulose, okay? The nature of bonding. Nature of bonding, right? Yes. I will try to discuss about the nature of bonding in the next class, okay. I will not discuss about here, but however, just remember one thing, the types of bond you find in the case of generally for our example is alpha 1,4, okay, or alpha 1,6, and one more we are having is beta 1,4 and beta 1,6, okay. And these bonds are actually called as the glycosidic bonds. We call them as the glycosidic bonds, right? So we will discuss about the nature of bonding shortly in the next class. So here the cellulose and the starch and the glycogen basically differs, okay, all of them differs in the nature of bonding. The difference between these molecules is that nature of bonding. And interestingly, one more point that we have to remember of the glycogen is that it is a branched polysaccharide. It is a branched polysaccharide. Yes. This is an important aspect that you have to remember. Unlike in the case of cellulose and starch, both of them are actually linear, okay? Linear in the sense just like a straight line. But however, glycogen is a branched, what we can say, polysaccharide, okay? There will be multiple branch points, okay? So branch points and all there again, as I said, it is advanced biology concept. Don't worry about that, okay? So glycogen is actually a branched polysaccharide, but however, cellulose and starch, both of them are actually linear polysaccharides, okay? Now similarly, one more we can say homopolymer in the case of polysaccharide is the inulin, which is made up of multiple units of fructose. It is mainly made up of multiple units of fructose. Remember this. So now when it comes to the chemical behavior or chemical property of the polysaccharides, you say that the right end of the chain is called as the reducing end and the left end of the chain is called as the non-reducing end, okay. Now non-reducing or reducing is mainly called because of treatment with one kind of reagent called as the Benedict's reagent, okay. Benedict's reagent, right? So now I hope you people might be remembering about the Benedict's reagent in your biology practicals, right? So in the first year practicals, you people might have done the test of biomolecules, for example protein test, okay, glucose test, and also you may have done some lipid test also. I don't think lipid test is present, but generally protein test and a carbohydrate test, that is the starch test, what you people will do. Okay, the appearance of what kind of color? Brick red color. Okay, the appearance of a brick red color indicates that it is actually a reducing, okay, it is actually a reducing sugar, right? This is what the conclusion actually you come into across. So it is mainly because the Benedict's sugar reduces that particular the Benedict's reagent reduces that particular sugar, okay, that is whatever sugar you are testing. So there are what we can say reducing sugars and non-reducing sugars also. Okay, so for example glucose is an example for reducing sugar, starch is also an example for reducing sugar, remember that. Yeah, so we can say that if you consider the chain of a polysaccharide, the right end is actually what we can say the left end is actually a non-reducing part, and the right end is actually what we can say reducing part over here. Okay, now if you consider starch for our discussion, it actually forms a helical structure. Okay, and helical in the sense a spring-like structure is called as the helical structure. Okay, it is called as the helical structure because the starch can behave in a helical way. It can actually bind it to iodine and it results in the formation of a blue color. Okay, but however cellulose can't hold iodine, okay, because or due to no helical structure formation. Okay, there is no formation of helical structures in the case of cellulose. Okay, so again these 2 points are important. Okay, they may ask you which of the following statement is true or not true, or regarding the cellulose. Okay, cellulose can't hold iodine. Why? They may ask. Okay, because it does not form any kind of helical structures. Now finally, let us look into some examples of the polysaccharides. Again, cellulose, as we have discussed, it is mainly found in the case of a plant cell wall, right? And there are some chemically complex modified sugars. Okay, for example glucosamine, and one more we are having is the N-acetyl glucosamine. Okay, so these are complex chemical sugars, okay, where there will be certain modifications done to the carbon moiety or carbon structure of that particular sugar. Okay, yes, again we can say that these are all modification of glucose only. And finally we find one more homopolymer. Again, chitin is also what? A homopolymer. Remember this, it is also homopolymer. Okay, it is a homopolymer of N-acetyl galactosamine. Right, remember this, it is a homopolymer of N-acetyl galactosamine. Okay, and chitin is mainly found in the exoskeleton of arthropods. At the same time, chitin is also found in the cell wall of fungi, right? It is also found in the cell wall of the fungi. So these are some of the important points about the polysaccharides that you people have to remember. So I guess we can start solving certain questions over here. Let me erase this. Okay, so let us discuss some of the questions again over here. Which of the following, or one of the feature of secondary metabolites is their function is not known, their roles in the physiological processes is not known, and they are useful to human welfare, all of the above. As I mentioned, we know all of their important functions. Okay, we don't know their function, what physiological process we don't know, but yet they are having some important value to the human welfare. Hence we can say all of the above statements are right. The next one, the molecular weight of the acid soluble pool ranges from the 200 to 2000 Daltons. Okay, 100 to 1000 Daltons, 18 to 800 Daltons. The last one we are having is the 100 to 10 to 100 Daltons. The answer we all know, it is 18 to 800 Daltons is the range of the acid soluble pool. Moving on, the only exception to the compounds in the terms of thousands of molecular weight. Okay, the only exception of the compound or the what we can say molecule which does not have thousands of molecular weight, but yet we call it as a biomacromolecule. That is what the question is, right? Lipids, proteins, carbohydrates, and nucleic acid. We all know it is actually lipids. Its weight does not increase, or it's not having more than 800. Okay, it does not exceed 800, but yet we call it as what? A biomacromolecule. Hence, it is the exception we are having. The next one, which of the following in the host organisms are known to have normal roles and functions? Okay, secondary metabolite, alkaloids, primary metabolites, none of the above, right? So we know in the host organisms, normal roles and functions are known to have for the primary metabolites, right? Primary metabolites are the substances which are known to have the roles in the case of host organisms. C is our right answer. The next one, micromolecules, a molecular weight does not exceed 2000 daltons, 1000 daltons, 800 daltons, 500 daltons. Okay, the answer we know it is 1000 daltons. Okay, the molecules, their molecular weight does not exceed than the 1000 daltons. Next question, which of the following is a drug? Okay, that is abrin, ricin, concanavalin, and next is the vinblastine. We all know these two are actually toxins, and this is a lectin, and vinblastine is known to be a drug over here. So D is our right answer. Match the following. Yes. So the column 1 and column 2, right? So let us look into the column 1. We are having alkaloids, toxins, lectins, and polymeric substances, and pigments, right? And next one we are having column 2, ricin, concanavalin A, and anthocyanins, codeine, and rubber, right? So for the fifth one, pigments, we know anthocyanins, so fifth one will come over here. And polymeric substances we are having is the rubber, so hence fourth number will come over here. Okay, lectins we are having concanavalin A, so third number will come over here. And in the case of ricin, so toxins we know it is ricin, so 2 number will come over here. Alkaloids, that is fourth, that is codeine. Morphine and codeine are called as the alkaloids, right? And hence the 1 number will come over here, right? So if you look over the options that they have mentioned, I can say that so the first one 4, second one is 2, and 3 is 2. Yes, exactly. I think we have got our answer. A option, I think it matches completely with what we have written over here, and exactly it is A is our right answer. Right, now moving on, which of the following is true about the lipids? They come in acid insoluble fraction. The grinding of the tissue will break the membrane. Okay, vesicles are not the result of grinding. They have a high molecular weight. Okay, so now they come in acid insoluble fraction. Okay, that is also true. And grinding the tissue will break the membrane, right? So we can say that both these two options are actually the right answer. Okay, so vesicles are not result of grinding. No, that is not true. And they have high molecular weight. Yes, that is also not true. So here probably while preparing the options, I might have what we can say missed out some aspect. So this question, it is completely invalid. Okay, I can say this is invalid, but yet let us consider that both these two options are correct. So probably the first option might have been they come in acid soluble fraction. Okay, so we can consider that if this had been the statement, then B would have been the answer. The grinding is not resulting in breaking of membrane. If that would have been the option, then A would have been answer. Okay, so I guess the mistake is on my side. Don't worry. Okay, probably during the typing way, I might have missed some vital point over here. Okay, so let us consider 8th question as invalid. Okay, generally in your NEET examination, such things will not happen. Okay, grace questions will not appear. Don't think in that particular way that you will get some grace questions. Never the case. Okay, they will make sure that all the questions with the respective options are proper. Okay, yeah. The next one, which of the following is an acid soluble pool. Okay, proteins, carbohydrates, cytoplasmic composition, lipids. We all know the cytoplasmic composition mainly belongs to acid soluble pool. The next question, which of the following is not strictly a macromolecule and yet found in the acid insoluble pool? Okay, not a macromolecule strictly speaking, but yet it is found in the acid insoluble pool. Proteins, carbohydrates, nucleic acids, and lipids. We all know it is what? Lipids is our answer. Okay, it is not a macromolecule strictly speaking, but yet it is found in the acid insoluble pool. Okay, and what is the reason? We know that. Yeah. Now, which of the following macromolecule makes the highest percentage of the cell? Okay, the first one you're having is the proteins, carbohydrate, next is lipids, and finally we are having nucleic acid, right? Around 10 to 15% we say that proteins are the major, what we can say, percentage of contribution when it comes to the cell. Right, we have mentioned this in the table that we have seen. Water makes around 70 to 90%, but proteins makes around 10 to 15%. The next question, the organelles of the cell come under acid soluble fraction, basic soluble fraction, acid insoluble fraction, and the next one we are having is the basic insoluble fraction, right? So we know that they come under the acid insoluble fraction, okay, right? The macromolecules and also the cell organelles, okay, they basically come in under the acid insoluble fraction. Okay, even the organelles, if you consider their structure in the cell chapter we have discussed, right? The cell membrane ultimately is made up of what? Lipids, right? Similarly, if you consider even the cell organelles, they are also at the end of the day they are made up of the macromolecules itself, right? So C is our right option. Next, which of the following is a branched polysaccharide? Starch, glycogen, cellulose, all of the above. The answer we know it is the glycogen is actually a branched polysaccharide. The next question, the fungi cell wall and exoskeleton of arthropods are made of cellulose, hemicellulose, N-acetyl glucosamine, and N-acetyl galactosamine. Okay, so the answer for this is actually the chitin, right? Yes, the answer over here is actually the chitin. But if we consider the sugar unit that is present in the chitin is actually the N-acetyl galactosamine, so we can say that D can be more appropriate. Okay, if they had given chitin as our answer, then chitin as the option, then chitin would have been the answer, right? But we have to consider the sugar unit that they have given over here. It is the N-acetyl galactosamine. Okay, it ultimately forms the chitin. So D is our right answer. Next, the most complex polysaccharides are the homopolymers, okay, heteropolymers. Next is the triheteropolymers, helical in structure. We can say that helical in structure is making the polysaccharide quite complex in nature. Okay. Next question, which of the following statement is true? Starch can hold iodine. The helical nature of cellulose is responsible for it to bind with iodine. Okay, glycogen is a storage material in the case of plants. The right end of the polysaccharide is a non-reducing end, right? So now you know that it is actually reducing end. The right end is actually reducing end. So this statement is not true. Glycogen is a storage material in the case of the animals, not in the case of plants. So even this is also not true. Okay, and the helical nature of cellulose is mainly responsible for it to bind with the iodine, right? So we know that cellulose does not contain complex helices, right? Or helix kind of structure. Hence, it cannot hold the iodine. Even this statement is also wrong. Starch can hold iodine. Exactly, A is our right answer. Now moving on. The polymer of fructose is starch, cellulose, glycogen, inulin. You know, answer is inulin. D is our answer. The next question, cellulose, glycogen, and starch, all of them are polymer of glucosamine, N-acetyl galactosamine, fructose, glucose. It is mainly polymer of glucose. D is our answer. It's a simple question. Cellulose cannot hold iodine because it is not helical. It shows a branched nature. It is a homopolymer. It does not have reducing end. The answer we all know, it is not helical in appearance. Okay, the structure does not show helical kind of complexes. The next statement, which of the following statement is true? Plant cell walls are made up of chitin, glucosamine is a chemically modified sugar, cellulose is a heteropolymer, glycogen is an unbranched homopolymer polysaccharide. Okay, so we know that it is actually a branched homopolymer, right? This is a wrong statement. Cellulose is actually a homopolymer, wrong statement again. And the glucosamine is a chemically modified sugar, exactly, it is the true statement. And the first, we all know the plant cell wall is mainly made up of cellulose. Okay, it is not chitin. Chitin in the case of the fungi and the exoskeleton of the arthropods, right? So even A is also the wrong answer. So I guess we have solved around 20 questions over here, and till now, if you consider, we have discussed around 40 questions. And you people, as I said, you can start solving more and more questions, and we will meet in the next class of the biomolecules, and we can discuss the further aspects, that is mainly the proteins and all, right? Thank you.